In this class, we are going to see some of the diseases which are serious on lentil crop causing economical yield loss. So the diseases which includes fusarial wilt caused by Fusarium oxysporum form of species lentil, and then Botrytis grey mold rot caused by Botrytis cinerea, and collar rot caused by Sclerotium ralphsi, root rot which is caused by Rhizoctonia solani or Macrophomina fasciolina or the perfect stage Rhizoctonia bataticola, and then the rust which is caused by Euromyces vca fabae, and anthracnose which is common in many of the vegetable crops, and here it is caused by Coltotrichum truncatum, and ascochyta blight which is serious on peas, chickpea as well as in lentils, and here this ascochyta which is caused by ascochyta lentis, and powdery mildew which is also a common disease on many of the vegetable as well as many of the field crops as well as horticultural crops. Here it is caused by the oidium type fungi Ericifae phyci. Let us see one by one about the diseases. First one is the fusarial wilt caused by Fusarium oxysporum, form of species lentil. The symptoms which includes yellowing, drying and drooping of wheel of the infected plants, besides wilting of all the leaflets are the primary symptom of this disease. The affected plant completely turn into yellow and completely die. In the longitudinal section of infected plant, which shows typical vascular discoloration in many of the other crops, is absent in this particular lentil crop, and the crop is mostly severely affected during the mid to late pod filling stage of the crop. When it is affected at the later stage, and it produces shriveled seeds within the pod, and the root system which may appear healthy, but the proliferation of root got reduced, and the nodulation percentage on the infected plant is also got reduced due to infection by fusarial wilt. It usually occur near the reproductive stage or flowering stage of the crop growth, and the other symptoms, allied symptoms due to this fusarial wilt, are seed rotting. So the pathogen Fusarium is known to produce three kinds of spores called macroconidia, microconidia and chlamydospores. These microconidia are oval to cylindrical in shape, hyaline in nature, single-celled. Normally it arises from the short conidiophores, while the macroconidia which are borne on the branched conidiophores, which are having thin-walled 3 to 5 septate fusoid with pointed tip at both the ends, and the chlamydospores which are rough walled or smooth walled, either terminal or intercalary, produce singly or in chain, which present in the infected plant debris as well as in the soil, serves as the primary source of inoculum. So these chlamydospores which can survive in the soil for a longer period even in the absence of the host. So the pathogen survive in the infected plant debris and serves as the primary source of inoculum. The debris may be remain viable or the pathogen remain active for the period of 3 to 4 years. Low soil temperature, warm and humid weather, and high and high humid weather are favoring the wilting symptom. The infection at a pre-pod stage is 100 if the infection is at pre-pod stage, the loss would be of 100%. If it is pre-harvest stage, then somewhat low yield loss of 67%, though it is very high compared to the infection during the pre-pod stage. The pre-harvest stage may be advantages. So to manage this disease, use the resistant varieties like Pant L4 or Pant L6 or Pant L8 and Nuri varieties, which will be resistant to this fusarium disease. And before sowing, you have to treat the seed with systemic fungicides like benomil, and then or biocontrol agents like Bacillus subtilis or Trichoderma viridi or Trichoderma harzianum, which will be helpful in very good seed establishment and protect the seeds, germinated seeds, from the infection of soil-borne inoculum. The organic matter amendment in the field that may enhance the antagonistic organism activity, that would also indirectly helpful in successful germination as well as growth of lentil plants. The summer plowing, and then if the disease is very severe, go with the crop rotation along with the non-host crop like cereals is also advisable to manage this disease. And then botrytis grey mold, which is caused by botrytis cinerea in North Indian fields, several in northern part of India, as well as several countries like Australia, Argentina, Nepal, Myanmar, Bangladesh, and Pakistan, etc. So this botrytis grey mold causes 70 to 80% yield loss when the environmental factors are conducive for the development. It affects all the above ground part of the plants. It appears especially on the lower foliage as discrete lesions, initially dark green and later turn into grayish brown, enlarge and coalesce, causes a blighting of entire leaf. Severely affected leaves may fall off, and the lesions on the stem causes girdling symptom. Girdling means literally encircling. So the lesion encircles the stem portion and causes complete rotting. So eventually the above part of the plant parts above the point of infection may got completely dried off, ultimately resulting in the death of the plants. And it spreads mainly through the seed-borne inoculum. Besides, the presence of sclerotia in the infected plant debris as well as in the alternate host helpful in spreading of this disease. Moderate temperature of 15 to 25 degree, high humidity, that is more than 95%, and high moisture especially during the flowering stage are favoring the development of this disease. To manage this disease, crop canopy management is very important. So before sowing, go treat the seeds with chemical fungicides like carbendazim or chlorothalonil or thiabendazole at the rate of 0.1%, which reduces the seed-borne inoculum. But besides, use of resistant lentil varieties like Panth L639, Panth L406 are also advisable. And then the collar rot disease. Collar rot, the name itself indicates the pathogen affects the plant at the collar region just above the ground level, and this is caused by Sclerotium ralphsi. The affected plants, due to infection at the basal portion, the affected plants turn yellow and rotting of the collar region. In severe case of infection, white mycelial growth could be seen on the affected parts with mustard like or rapeseed like brown regular sclerotia. These infected plants are usually seen in patches in the field. So sufficient soil moisture with high temperature of 28 to 30 degrees Celsius favors the development of this disease, and the presence of sclerotia in the infected plant debris as well as in the soil serves as the primary source of inoculum. So to manage this disease, treat the seeds before sowing with the fungicides like carboxin, or if the infection occurs at the later stage or during the crop growth, then drench the infected plant with the carboxin at the rate of 0.1%. Besides, you can go for application of biocontrol agents like glycolidium virens, trichoderma harzianum, Bacillus subtilis are also much helpful and act effectively against the pathogen Sclerotium ralphsi. And then the root rot, which is caused by dry Rhizoctonia solani as well as Rhizoctonia bataticola, which is the perfect stage of Macrophylla fasciolina. So the Rhizoctonia solani as like that of the other pulses. Rhizoctonia solani causes wet root rot, while the Rhizoctonia bataticola or Macrophomina fasciolina that causes dry root rot symptom. So to manage this root rot pathogens, better go for a soil application of biocontrol agents like Glycolidium virens, Trichoderma harzianum, Trichoderma lingorum, Trichoderma asperellum, etc. And before sowing, for the chemical management, before sowing, treat the seeds with the carbendazim at the rate of 1 gram per kg, or during the plant growth if the symptom occurs, apply carbendazim at the rate of 0.1% to reduce the incidence. Especially it has to be applied in the drenching method. And then the next important disease is rust disease, which is caused by Euromyces vca fabae. Like that of the other crops, here also it produces raised pustules on the surface of the leaf. Here, yellowish white pycnia as well as Asia cups are produced on the lower surface of the leaflets as well as on the pods. So this Euromyces Vicia fabae is a autoceous rust and a macrocyclic rust which is having all the 5 stages of rust. So among the 5 stages, this pycnia as well as Asia, which are producing yellowish white in color fruiting bodies, especially on the lower surface of the leaf. These pustules, iridosauri pustules, are oval to circular, which may be produced on either side of the leaf as well as on the stem as well as on the pods. So these iridosauri are the pustules which coalesce together and form a larger infection on larger proportion of leaf area, leading to drying of leaves. This iridosauri, upon maturation, converted into teleosauri. This teleosauri occurs late in the season, which are dark brown or black, and which also present on the branches of stem as well as on the leaf. In severe infection, the leaves turn dry and they may fall off. The affected plant dries without producing any pods or seeds. Even if it is produced, the seeds will be shriveled in nature. So, as we discussed earlier, this is pathogen Euromyces vca fabae is an autoceous rust and which is having a pycnial as well as a axial stage on the undersurface of the lower leaves. Then iridosauri as well as teleosauri on the upper surface of the leaf. The iridospores, which are ellipsoidal when finely accumulated, while teleospores are also ellipsoidal, chestnut brown in color but with the smooth wall. High humidity, cloudy and drizzling weather which favors the development of disease, and the temperature of 20 to 22 degree favors the development. And the disease occurs especially during the flowering or early pod stage. In order to manage this disease, you can go for using the resistant varieties like Pant 639, Pant L406, Pant L6, Pant L7 and Pant L8 type of varieties can be used, or else a foliar application of carboxin or in combination with the mangoseb is highly advisable to reduce the incidence. Ascochyta blight caused by Ascochyta lentis. This Ascochyta, which is a serious pathogen on pea, chickpea as well as on lentils. It initially produces irregularly shaped tan brown lesions on the leaves as well as on the petiole, stem and pods, even on the seeds. Later, the infection get enlarged with a dark brown margin and grey center with numerous black dots. These black dots which represents the pycnidia of the fungi. Such deep-seated tan brown, dark brown lesions on the stem which causes girdling symptom, and the plant parts above the point of infection are dried and the plant may break off at the point of infection, leading to death of tissues or death of the plants. Even the seeds are shriveled, completely discolored, and in severe case of infection you may see the presence of white mycelial growth as well as the formation of pycnidia on the seeds. The pycnidia formed on the infected plants, which contains numerous cylindrical, straight or rarely curved, rounded ended conidia, and this conidia is responsible for severe spread of the disease. The teleomorph or the perfect stage of the fungi is Deidimella lentis, which is observed for a first time on the wintered lentil straw, which produced fertile pseudothecia. So pseudothecia, which in turn contain ascus and ascospores. So this ascospores may serve as the primary source of inoculum, followed by the airborne conidia, which serves as secondary source of inoculum. The seed borne as well as the seed transmitted nature of this disease leading to severe incidents in the field conditions. Cool, moist weather and the leaf wetness for a period of 24 to 48 hours with the temperature range of 50 to 68 degree Fahrenheit are favoring the disease development. Maximum incidence of this disease was noticed during at 59 degree Fahrenheit temperature. So when the disease is very severe, as we discussed earlier, the pathogen may remain active on the infected plant debris and hence go for crop rotation with the non-host crop. Which will be helpful in reducing the incidence. So it is advisable to grow lentils once in 4 years. So use of certified as well as disease-free seed are also recommended. Early sowing of lentils which will escape the moist weather at the time of harvest. So fungicides such as carbendazim, thiobendazole are recommended for the management of this disease at the rate of 0.1%. Anthracnose, which is also a common disease in many of the vegetable crops. Here, the anthracnose, the non-lentil, which is caused by Colletotrichum truncatum. The symptoms produced by this pathogen is also similar to that of Ascochyta blight. The leaflet lesions are grey center, dark brown margin, and black dots at the center portions. So in the case of Ascochyta blight, those black dots which represents pycnidia of the fungi. But in this case, the disease caused by Colletotrichum, these black dots which represents acerulae of the fungi. This deep-seated sunken lesions formed on the stem may cause girdling symptom, and it also produces lesions on the parts as well as on seeds. So affected seeds may become discolored or shriveled or malformed. So the pathogen which remains active on the infected plant debris as a sexual stage as well as on the asexual fruiting body acerulae. So these conidia from this acerulae serves as the severe spread of this disease. To manage this disease, better go for field sanitation, that is collection and destruction of plant debris, which will reduce the field borne inoculum. And during the cropping season, you can go for spraying with the chlorothalonil at the rate of 0.2%. And then sclerotinia stem rot, which is caused by Sclerotinia sclerotiarum. This sclerotinia, which produces initially lesions at the basal portion of the stem, causing girdling symptom on the stem, and also produces chlorotic leaves at the top, and the entire plant will become completely wilted. During the moisture period, it produces cottony white growth on the affected stem as well as on the leaves. So the sclerotia that may remain active or remain long in the plant debris as well as in the soil. This fungus is known to produce apothecia as the sexual ascocarp, so which in turn contains ascus and ascospores. This apothecia can persist in the soil for a longer period of 5 to 7 years. So the presence of sclerotia as well as presence of apothecia are responsible for the primary responsible for the primary spread of this disease, and the secondary spread is also due to the water and the favorable environmental conditions. Cool, wet condition and presence of dense canopy are favoring the development of disease. In order to control this disease, you can go for spraying with azoxystrobin at the rate of 0.1%. And then stem phylum blight, which is caused by stem phylum species, it produces infections on the leaves. So affected leaves which turn yellow and the entire leaf become dried and the entire plant become defoliated due to severe infection. Complete defoliation occurs within a shorter period, that is a week of period after the infection, that is sufficient for complete defoliation. And the fungus sporulate enough on the live as well as infected leaves and produces blackish or grayish mold of mycelia as well as conidia. So the infected plant debris presence in the soil serves as the primary source of inoculum, and airborne conidia responsible for the secondary spread. To manage this disease, we can go for spraying with the chlorothalonil at the rate of 0.1% or mangoseb at 0.2%. That would be highly advisable to reduce the incidence at the field level. And then powdery mildew. Here in the case of lentil, the powdery mildew is caused by the oidium type of fungi, that is Ericifae PC or Pisces, which is causing oidium type infection, that is it produces grayish or dull white powdery patches or powdery spots on the upper surface of the leaf. Under severe conditions, it covers the entire leaf, leading to the drying of the affected leaves. The affected dried leaves may fall on the ground. In the affected dried leaves, the fungus produces the sexual ascocarp called chasmothium. So the fungus which overwinters in this plant debris as well as in the on the alternate host. Generally, powdery mildew is favored by the warm, humid weather, and cool nights will also be helpful for the further spread of this disease. Use the resistant varieties, and the infection will be much severe if the plant is sown late. So early sowing is advised to avoid or to escape from the severe infection of this powdery mildew, and using the overhead sprinkler, which will helpful in washing the surface borne powdery patches. And the plant the crop at the early possible, so that the severe infection can be escaped. And frequent application of sulfur, which would be helpful in reducing the infection. So besides the studied fungal diseases, there are few viral diseases are also important on lentil crop. That is alfalfa mosaic virus, which causes necrosis on the tip of the plant, which leads to twisting and deformation of the leaves, while this cucumber mosaic virus, which causes chlorosis of the affected leaves, yellowing and stunting of entire plants. The bean yellow mosaic virus, which also causes chlorosis and it causes leaf malformation besides stunted growth of the plant. And then pea seed-borne mosaic virus, which causes chlorosis on the new shoots and mottling pattern on the leaves and shoot tip necrosis as like that of alfalfa mosaic virus, and causing stunted growth of the plant. So all these four types of viruses are transmitted by the insect vector, that is aphid vector Mysis persicae as well as Aphis craxivora. So to control this insect vector, spraying of methyl dimethoate at the rate of 500 ml per hectare or any other important or effective insecticide against the sucking pest can be explored for the management of this aphids. Thank you.